and then we'll do sharing, and you should see yourselves and a presentation here, which we can look at. And to a degree, I'll try and put this in context of small-scale processing, but quite a lot of the suppliers who give you the malt, who process it and so on, are doing so on a very large scale, and their customers tend to be, in the majority, the big breweries. So if we're looking for something which is very specialised or focused on small scale, then there are ways to adapt, but sometimes you have to accept the standard material they have online. And what we're going to cover is what is the malting process, a look at barley and malt, their features and differences and such, the actual process stage by stage, the cultivation of barley to a degree, because there are features as to the cultivation characteristics, what the farm does, how it affects the final product, the varieties of malt, but we're not going to go through one after the other, but we'll mention a few which are particularly interesting to the small scale production and how they relate to the malt types. Specifications of malt are more important than specifications of barley to the brewer. As a brewer, the specifications of malt are more important than the specifications of barley to the brewer, because you're looking at the final product you put in your mash tun. The barley itself is, yeah, it's got characteristics, but it's the maltster who is particularly keen to see those specifications. Look at the malt analysis. What do we characterize the malt quality by? So those are the areas. We're going to think about, well, malting as a slightly divorced germination process of a seed, and that is standard in sense of horticulture. You're taking seeds. Normally they would go in the soil, they would grow, but we're taking them and putting them in a controlled condition, allowing them to germinate, go through their reactions internally, and then make them of a condition that we can use to extract sugar. And that sugar and the other things as well that are in there will go into the brew. We've got to control conditions, and the very important ones are moisture during the processing of the malting, the temperature of the maltings, and how long we leave each stage. And we can change the characteristics of the malt by varying how much hydration there is, moisture in the grain, how long, what temperature we put the grain to germinate and so on, and the time that it stays. If we go back in long times, hundreds of years maybe, this was done almost by eye. People didn't really measure things. And some of that information has translated to today. So you will find maltsters operate on almost a historic basis, but monitor it and then control if there is variations. And I think it's worth saying right at the beginning, there's always variations from season to season. And it's very much more perhaps apparent in hops, where the characteristics of what you're harvesting are susceptible to climate or weather conditions and such, and you've got to keep a track of that. Now, the result of malting is to give you the grain, which you can then use for mashing. And the mashing process gives you, in the end, the sugars that you use for fermenting. So we've got to put it in context of the overall brewing process. And there's a bit of background in the sense of knowing what extract is, knowing what enzymes are, and how colour and flavour come out and are processed into the beer. Now, the support material we can look at on that, which will provide you the background to extract, which in a way is what you deal with every day and measure when you're preparing your wort, keeping the enzymes active through the right conditions of temperature and so on. And then the colour and the flavour are things which are biochemically focused and can vary. And it's probably important to say there are positive levels of colour and negative levels of colour. If you're producing a particular beer, you might want high levels or low levels of colour. You might want particular flavours and not others. And there are some flavours produced in maltings like dimethyl sulfide, which you don't particularly want. So controlling them is not just enhancing, it's also limiting. Now, two differences, barley and malt. Well, they look pretty much the same. they're both dried products. They've got grain structure. You can see that they are fairly consistent in both of these, but there are differences. Barley and malt, and when we put them together, what we find is that in the barley, which comes into the maltings, we've still got some of the farm material still on there. Need to remove that or wash it, not wash it off, but account for it. We would expect the farm to manage its applications of pesticides, let's for example say, so as to not release a product which has got extensive levels of them. Barley is quite a hard product. If you chew it, it's really easy to break your teeth because it's so hard. It's a really compact package of starch. If we just put it into a mash tun, we won't get a lot of sugar out because it's mostly starch, which needs to be broken down and digested into sugar. The fermentability is pretty poor because it is mostly starch, and starch can't be fermented by yeast. If we do get it to dissolve, then it'll be very thick. It's very viscous because starch is a big molecule. It's not easy to sort of molecularly move, and therefore we're going to find problems in pumping it, in filtering it. There may be, if we just use barley without germinating it, not enough protein in the wort. And we need protein in the wort to digest 2 amino acids for yeast to grow. So there's a range of things which we've got to maintain and control the digestion, if necessary, of the grain. The flavour, if you chew it, is grainy in a way. It's starchy, so that's not very sweet. And there may well, depending on the variety, be a few slightly harsh or phenolic characters. If you just crushed up and dissolved barley, there wouldn't be a lot of colour. There's a bit in the husk, but it's not going to dissolve very well. The wort would be cloudy because of the undissolved or the undigested starch. And there wouldn't be a great deal of enzyme levels, although there is some, and it'll begin to digest the starch depending on the temperature. So being aware of all of that says, well, we can't easily make beer with just barley. That being said, historically, people did because they had conditions which allowed the barley to germinate a little bit, grow a little bit, or there were microorganisms which would digest it. But today, when we're under pressure of time and so on, we can't just use barley. We have to convert it into malt. And malt features are, in a way, the opposite of what we've just said in many cases. We should have the agrochemicals washed off. It should be millable, it should be soft, and therefore we can crush it and it's friable. So it's sort of crunchy, but it's not solid hard. The extract, the amount of sugar we get out should be high. Arguably, the more efficient your malting system is, the higher the extract you'll get, although you balance that between the efficiency of extract and the colour or the flavour as well. When you digest it and put it in the mash tun, you're going to get fermentable product, fermentable wort, because there is sugar in there, simple sugars, glucose, maltose and so on. They will be fermentable. There'll still be some large-scale polysaccharides, and they will be probably unfermentable, and that provides a different character to the beer. Viscosity will be lowered. There'll still be some protein, or in fact a reasonable amount of protein, enough to encourage the yeast to grow. Flavour, malty, okay. In a way that's different than grainy. It'll be sweeter. It'll have more of a toasty, bready character. Colour, desirable according to the variety of malt. When it's dissolved, they generally produce clear wort, and there'll be enzymes in there which will digest the starch into sugar. So there'll be a little bit of digestion ready in the malt, but not a great deal. Now, we're looking at barley coming from farm conditions, growing and so on. Why we use barley? Well, there are these reasons, but I'll just aside say we can also use wheat, we can also use sorghum and a variety of other cereals, but barley particularly because it's got a good external covering, a husk, and that means we can use it as a filter. And when we filter through in the mash tun, we get a clear wort. It leaves the solid material behind. Now, what is the solid material? The solid material is the remains of the cell material, the barley material, the husks, and so on, but it's also precipitated protein and polyphenols. So it's not just a crude filter, it's a molecular filter which adsorbs the chemicals and the materials we don't want that would make the wort in the end cloudy. It's got a high starch content. That gives a lot of sugar for fermentation when we digest it in the mash. So the high starch is a very important element of it. You can get different grains which have more or less starch per grain, and the 2 and the 6 row varieties have different proportions of those. In a sense, if you have a big grain, you're going to have a lot of starch inside. You're not going to have so much husk on the outside in proportion. That
So
has an
you don't have a very good filter bed if you use, for example, the 2 row instead of 6 row. Yeah, the 6 row has more husk, so it's better for the filter. Okay, for the lauter tun.
Indeed, exactly right. So you've got more husk, which gives a better filter. Yeah. Now, inside, as we'll look at, the cells of the barley grain are degradable by enzymes. Before the enzymes act, the starch is surrounded by cell walls and protein they need digesting before the starch can be dissolved and then digested. So it's very important that we've got barley varieties with the right enzymes in them. And when we do the mash, which we'll talk about another time, they have the right temperature control in the mash, progressively or at the balanced temperature. Those enzymes convert starch to sugar, but also there are enzymes, protease enzymes, which digest proteins, and glucanases, which digest glucans. So there's a mixture: amylases for starch, proteases for proteins, glucanases for glucans. We want to result in a breakdown of, in the mash, the starch to sugar. We don't want that much to happen in the malting process. We want it in the mashing. But in the malting, we will have some breakdown, or a fairly extensive breakdown, around the grains of starch. We'll have an extensive breakdown of proteins and also of the cell walls, which is the glucan material. It's also worth saying that barley is both freely available worldwide, it's transportable internationally, and it grows in many climates. And we'll talk about that in a minute, but you can have it, produced locally. And again, if you were a small scale producer, you could associate with a farm to produce your malt, as long as you can get it to a malters to do that. Now, phases, and when we're thinking of examination levels and so on, this is what they like to make sure that you're fully aware of, that not just the different stages, but the features of each stage, which we will look through. We're looking at steeping, which is where the grains of barley are soaked in water, in liquor, and that hydrates them and activates the enzymes, activates the germination, which is the main part of the maltings. So first off, a day or two steeping, soaking the grains, then we transfer it into a germinating vessel, goes and gradually produces initial shoots, initial roots. Once that's developed and we know that the enzymes are in the right balance, and we can move it and dry it so it's ready to process and sell as malt. So steeping, germinating and kilning, in a way it's fairly straightforward to get them in your mind, knowing what's happening at each stage. But let's look at the details. But to produce malt, the barley we want is to be of good condition. So this one here in the middle has all intact grains, and they're not infected with any disease like the two on the right. On this one in the middle, we've got mildew, which grows on leaves. Now, it stunts the growth a little bit, and because it's often held in the soil, it's often on the lower leaves. So as the plant grows higher, it'll probably grow away from mildew. What we really, really don't want is these grains here, a fusarium infection of head blight, where the fungus in this case digests the actual grains themselves. And that's obviously going to lead to a loss of, well, extract, a loss of starch. It'll also produce, if it's fusarium, very often a toxin. Those toxins are hazardous if you're going to process a food product. So the person producing on the farm, but also monitoring it in the maltings, is going to be assessing for that character. So we want the right variety. There are a whole range of varieties we need to look at a little bit. We need water, we need oxygen to allow the respiration to occur in the plant, in the seeds. We need the right temperature, reasonable 20+ degrees maybe. We need the right time, which we've just mentioned and before. So let's go through by stages, that's steeping, germinating and kilning. First off, barley has got to go from its storage hydration, 12%, to enough of a hydration to activate and solubilize the enzymes to then do the initial breakdown of the glucan and the protein. And that initiates germination. So we've got to put it in some water in order for this to happen. Germination, control condition, it sprouts and grows, and kilning, we dry it. So we'll come back to each of those specifications. But let's look at steeping. Here we've got steeping vessels. They are very large and they're going to hold maybe 5, 10, maybe even 20 tons of grain. So they've got to be large, heavy construction, really, really robust. And there's going to be a processing whereby the grains enter and then the water enters and they gradually mix or have a mechanical mixing system, as you can sort of see there. These are figures the Institute questions often ask. What is the hydration level of a grain before steeping and after? 12 generally, that's a sort of standard for farm release or reception at the maltings. 45-ish is going to be what's target to get germination going. And the germination needs to be uniform. So it's no good just having the grains stuck at the bottom and a lot of water on top. Some of those grains won't have contact enough with the water to absorb it. So the mechanical mixing needs to be such. Some of that mechanical mixing can be by blowing air through it, because that's going to provide oxygen. To some degree, we might want specialist malts such as crystal, where it's kilned while it's still fresh, and that might need 48%. And we've got not just soaking in water. Very often the steeping involves draining the water out and refilling it. That's quite good because it washes the grain and it provides a replenishment if there are any things released from the grain which could inhibit the germination. And when you think that grains have been sort of stored for a period of time, they've been dormant, there are things in grain which enhance dormancy. So we want to get rid of those. And emptying filling will also bring more oxygen in. So the process of the stages of soaking, draining, leaving to dry, soaking again, blowing the oxygen or the air through, are controlled carefully by the maltster.
And also removes the carbon dioxide. Okay. Yeah.
A very good point. Yes, it gets rid of carbon dioxide that's built up from the germination reactions. Yeah. This would be the staging process of hours. So 8 hours, water's drained out, left for another 8 hours or so, and then water is replaced and drained and replaced and so on. And the moisture increases progressively through that. And we can see a diagram here of a steeping vessel. We've seen an image of one, but there are conveyors to drop the barley in. There's probes to monitor. There's an overflow in case the liquor does get too high, and there's an extraction which is going to be a conveyor to take the steeped grains into the germinating vessel. So again, what they might do in an exam question would be label the diagram, so you know what's where and what they're doing. Now, the beginning of germination requires gibberellic acid as a hormone, which it initiates the synthesis of the enzymes. And there's different enzymes, particularly the amylase, but it's the protease and the beta glucanase, which is going to digest the cell walls and the protein.
So also I read the xylanase, which degrades the arabinoxylans. I think it's low proportions as the big glucans in the cell walls. I don't know if I'm right, but yeah, I think something like that. About the degrade. Yeah.
Silicates.
Okay. Silicate.
Yeah. So they'll be in the cell walls and are things which can have an impact on the process and on flavour and polyphenols and so on.
Yeah.
Temperature and humidity are big important controls on this. And during germination, the whole body of the grains needs to be mixed so as to, well, mix the oxygen in, vent out the carbon dioxide, avoid the rootlets sticking together so it gives space between the grains. And again, different varieties and different conditions will give different degrees of germination. And what we want, or the maltster wants, is even germination so that all the grains are at the same stage. And a visualization of a germination vessel is fairly, in a sense, straightforward. It's got a working floor with the germinating malt on it, but it's got an underfloor which is going to direct air in through the perforated plates so that it can be stirred and mixed with the air, venting out the carbon dioxide, bringing in oxygen continuously. These tend to be fairly, let's say, modern. You know, we'll look at the sort of traditional way, which was just on a floor, but these are very mechanized. And I'm just going to run through this kind of image of one quickly. An image of the grains. No, there we are. So these vessels tend to be these things, which are taking tons and tons of grain at one go. They're rotating, and that mixes them, and you've got the spirals here, which will actually move the grains themselves. So that's what we're thinking of in actual view of it. So let's get back to what's happening inside the grains themselves. This is also a nice diagram for questions for you to demonstrate your knowledge of the component parts of a grain, where we've got the husk surrounding it, the endosperm full of the starch grains, the embryo, which is the nascent shoot and root.
I thought the questions are multiple choice. I didn't know that we have to,
They will be multiple choice, but what they will do is give you a picture and say, what is A, B, C, D? Or
okay.
Yeah, they can easily make this into a multiple choice, Tassos. No problem. So there is an embryo. The scutellum and the alurone are important layers. The alurone is where the enzymes are made and released into the endosperm. The scutellum is a boundary of mostly permeable cells to allow the results of the digestion. You know, if the plant was growing, it would use the sugars digested from starch through it and into the embryo, which it would use as the nutrients and the energy to grow. So that's the sort of controlling barrier layer. The endosperm has all the grains of starch, and the husk is a protective layer around the outside, which is dead, but it does have an opening whereby the water can come in at the right point. These are the enzymes which are involved, various, well, glucanases, we've said, amylases, xylanases, because they're involved in the cell wall, and proteases and lipoxygenases, which are going to digest some lipids. These component parts, again, are bits that you could be questioned on. The tester is part of the husk, but it's the sort of slightly inner layer. That's a view round showing the cells, a sectional view showing the cells in the endosperm particularly. And if you view with a high power microscope, then what you would find is that the grains of starch would be embedded in, well, all these cells of the endosperm. These are the scutellum cells or the alurone cells. No, I don't think they're the alurone cells, which will produce the enzymes. Oh, there we are, alurone, endosperm. And then the protein you can see on the surface sort of surrounding these grains. Those are the grains of starch. The protein sort of embeds around that and holds them stable, limits the digestion while this thing is in dormancy. And cell walls you can see there as well. So as time goes on, they get digested, and the beginning of the starch digestion is evident by little holes that appear in the starch grains. Now, although they are tiny holes, what will have happened is that the enzymes will have started to digest below those. So that will partly be hollow almost if it's advanced in its digestion. But here, while it's still germinating at the beginning, you don't have that starch digestion, but you do have the protein digestion. So the starch grains become fairly open. And you can see the difference in the actual grains themselves as they germinate. The roots and shoots, well, particularly the roots, grow out from that. And you can see within a few days, you've got quite extensive germination, and it should be even germination as well. So we want these things in the processing. Modification is a term used not just instead of germination, but for the digestion of the starch. So it's the, digestion of the protein and the cell walls. So if it's well modified, it will have good digestion, and particularly of the cell walls, and the starch will be readily available when you crush it and put it into the mash tun. The brewer wants to have nice, tight, readily crushable grains, friable grains, which when they are broken, they release the starch grains out into the mash. You don't want more protein to be still stuck there. You don't want the cell walls to be present. Now, kilning is where we dry it, or the maltster dries the grains. At the end of the germination process, it's stopped. The whole process is heated up, and that will evaporate some of the water down to very low levels. It was 11 at the beginning, but now it's going to be 3 or 4, 5, 6%, and it can be stored at that temperature. Oh, sorry, that level of humidity or water, and will not encourage the growth of microorganisms on it, and it will not encourage degradation internally of the malt itself. Kilning also can increase the colour and change the flavours. The big important thing is the processing of the heat. Now, it's done by passing heated air, initially at a low temperature and progressively up to 100 or more degrees C. And that air pressure or air temperature is very important to regulate, because if it comes through too high a temperature at the beginning, when the grain has a lot of water in it, there will be reactions within the system that will be negative in flavour and will degrade the enzymes, so you won't get a very good mash extract.
And of course, we have the stew, okay? Because in the I remember exactly the name. It's adjunct. It's adjunct. Yeah, they stew it. Yeah, it's I don't know how they're doing it, but it's like stew, you know.
Yeah. So you're talking about an adjunct rather than
roast barley. I think it's roast barley. Yeah.
Okay, we'll talk about roasting, because you can take the grains when they've finished the germination and roast them, or yes, you can just take barley and roast it, and it doesn't go through any germination at all.
Okay.
Mm-hmm. Now, compared to the steeping and the germination vessel and so on, we've got a bit of a more complicated system here, but this would be, depends whether it's a germinating kilning vessel, whether it's a kiln itself. So if it was a kiln on its own, you would have the green malt coming in, still wet, goes onto the grain bed, and then there's hot water coming, or sorry, hot air coming through from the heating coils here with a fan through the perforated plates, circulating round through a heat exchanger. So it's a matter of controlling those processes of the fan speed, the heat of the coil, and the circulatory system, not having the bed too thick to limit or inhibit the flow of the air through it. And of course, when it's discharged, it's taken away by a conveyor as well. So at the beginning of kilning, it's fairly low temperature, 55 or so. That cools, cool drying, and then it goes up to 95 to 100 for ale malts, bit lower for lager malts because we don't want so much colour, and a little bit more enzyme activity. But it could be higher temperature for darker malts. Generally low colour, but the higher temperature gives more coloured malts because reaction between proteins and sugars, glucose and so on, gives these melanoid in darker colours. If we kiln it wet before drying it, just kiln it straight away, then we get crystal malt. And the crystal malt has a different sort of composition because you will have quite a lot of coloured compounds being produced. And crystal malt has its own sort of caramelly character, which is quite important in some beers. And as you've just said, roast barley is non-malted, so it just goes into the kiln as standard barley, 11%.
Yeah,
and
I was saying before for the wet killing, it's something like the stew. You know, it's yeah, it's got high moisture, and they heat it. So yeah, and they produce the crystal malt. Yeah, it's something like that.
Now, it's interesting you say that. One of the things it took me a while to realise when brewing that I often just ordered crystal malt and got a particular crystal malt, and I never realised that you could get, and maybe years ago you couldn't get different types of crystal malt, which would be kilned at different temperatures, and they give different flavours. Not very different, but it's worth being aware of. You can get light crystal, medium, and high crystal, which give, suitable for certain styles of beer. And yeah, it might take a day to complete.
Okay.
Now, from the point of view of managing maltings, these stages are present. It's worth knowing these again for the institute because they might give you a diagram like this and say, what's this thing here, or what's doing that? Obviously, offloading from the farm into large-scale bins, goes through a screen because they're trying to take out stones and soil and bits and pieces like that. Dried off from 11% down, maybe a bit. Storage again, then through another screen into eventually the hopper to put in the steep. So it doesn't come straight from the farm into the steep. It has to be processed, checked, make sure that there's no foreign or reduced foreign materials, and that the drying is to the set amount. In the steeping vessel, then you've got green malt going in, you've got the circulatory process, which we talked about how long it takes, draining it, filling it again, and so on. And eventually the goes into the germinating vessels and into the kiln. So there's movements all the way through, and there's movements of air, there's movements of the material and of water and drying. So sort of knowing which stage has which of those specifications is, if you're a maltster, very critical. If you're a brewer, okay, good to know. The exam might cover them, and you might, if you at some point in time say, I would like to get a malt of this specification just for me. Once it's kilned, then it's got to be tested and stored ready for dispatch and sale. So the rootlets have to come off. That's important. We're not wanting to mash those. They'd cause problems in it, and particularly in the drainage through. But once the rootlets have been removed by an aspirator, then and blown away, then the whole batch is stored, goes through a screen again. All the small bits are blown away and stays in the loading bins before it's dispensed either into large scale bags or small scale for small breweries, 25 kilograms. Eventually, it might actually go through into a hopper on a brewery, but most small scale buy in bags. Yeah. And here we had a quick look at steeping vessel. This is an example of one that's fairly full, but it'll be, soaking with water. The steeping will be spraying water on as it mixes with the grain. The germinating kilning vessel will be a big expanse of floor with perforated plates, slots generally, and it will fill from the conveyors, spread out across the floor. Then this will rotate through to mix them, and eventually it'll be kilned, because the kilning will be integral to the whole system. Years ago, there were two approaches that were traditional. One was floor malting, which we'll look at in a minute. The other one was what's known as salad-in boxes, which is a long trough of a box with these screw threads going through it. And they would move up and down mechanically and mix the whole system, and there would be a perforated floor. So it's almost the same as a germinating kilning vessel. It only germinates and it has the same sort of mechanics, but it's a smaller scale. Eventually, if the conditions are right, then it goes through the conveyor and it just takes all the grain away. They're called JK or germinating kilning vessels, GKV, because they do both processes in one vessel, very automated, but somebody has to keep a control of it. And the laboratory tests, so you know when everything's ready, and it will look, it's a separate time about what the specifications are. If your brewery is big enough, you might be able to put 50 tons through and say that's my malt, and they store it for the year and you use it progressively. And in the UK, these are some of the names of the suppliers, but they will send overseas as well. That's just a view of one of their systems in place. But floor maltings, which is where traditionally years ago, and still in some parts of the UK, particularly in distilleries, they throw the grain on the floor and then they keep moving it around physically. So you have a layer which is 1/4 of a metre deep and turned at regular intervals to provide the oxygen, remove the carbon dioxide, keep it even and not clumping up. Temperature may be no more than 15 degrees. It germinates there, and really this was seasonal years ago, when they could only do it in the autumn and spring because it was too hot in the summer and too cold in the winter. And this is a typical view of a floor malting. It does have some louvers in the window, in the sides as windows, but somebody's got to shovel this stuff and also separate it by pulling through a rake. And eventually it goes into a kiln, which is a similar room, which has perforated floors. But not much malt's produced this way. It's not very economic. It's very expensive. It's specialist. And some breweries say that's the malt I want because it is, well, it is different. And to be honest, not to be honest, but I have looked at the microbiology of these and compared malt from salad-in boxes, GKVs and floor maltings, and found there is a difference in the lactic acid bacteria. There's more in the floor maltings, as maybe you might imagine, and different types. And those lactic acid bacteria do help in the digestion, but also in controlling the presence of other microorganisms. So I would say, from my experience, there is the claim that this is a little bit purer malt may well be true. But I don't think I've seen a big sort of big detailed research study on that. Jumping back to barley, grown around the world, we can see all places, where it's grown in temperate to even tropical regions. So in a sense, every continent will have its own supply to different degrees. And in the UK, it's going to be in usually the east of the UK and Scotland, because it's a little bit drier and it's not therefore going to develop so much of mold to grow on it. Certain soil types are better, and it's got, needs certain access to distribution and so on, transport. Very important, as you mentioned, Tassos, the difference between 2-row and 6-row. These are commonly available. 2-row has only 2 rows of the barley opposite each other. 6-row has 6 all the way round.
Yeah, only the 2 rows, only 2 flowers are fertile. Okay, and the 6 rows, the 6 rows are fertile. 6 flowers are fertile.
Yes.
Yeah. Okay.
If you look down on the top, you can see it like this.
Yeah.
And you can see there's 1, 2, 3 rows there and 2 more at the back. 3 more at the back. What is worth saying, the 2-row barley just has 2 fully grown corns, but there are 4 sterile corns as well, so they poke out at the side. So yes, you can see them here. That there and those there are sterile. So there are still 6, but only 2 grow out. But,
and I think it's in 2-row, it's a little more bigger, give us more yield. Okay. Yeah.
Correct. There's more yield because there's more starch, endosperm size in each grain.
Yeah. Okay.
But some people prefer, as you've said, with the lautering, to have more husk. So therefore, you might have a mixture.
And it's more economical, I think, the 6-row for the breweries. It's more, yeah. Yep. I think it's more cheaper.
Yep. So you can see also a different shape on the 6-row. They're slightly twisted because of being crunched together, whereas the 2-row can grow with more space. There's also winter and spring. So we've got winter barley being planted in September, October, spring barley in February, March. So winter and spring, harvested at slightly different times. And it depends on preferences to a degree of varieties. You'll get a greater yield from the winter, but you might get different character, well, you would get different character in the spring. So different breweries have their own preferences in the choice of those. If we look in detail at the grains, the barley grains, we've got other things which the Institute exam questions might like you to know. Like any plants, flowers, there will be anthers producing pollen. There will be stigmas which take the pollen and get pollinated. There's these pellia, which are the outside parts, the resistant parts and so on, and inside there's an ovary. So that all develops into the final grain. When the grains are growing, we will have various options that we could use as well, or that farmers will grow, particularly wheat, because that's a very required food product, but oilseed rape and fuel crops and so on. Malted barley, compared to these, could be said to be high risk because you've got specifications and quality, particularly of the enzyme level and the protein level, which need to be met. So if you're going to try and grow malting barley, which you would give a premium price, you've got to manage it very carefully and achieve that. If you don't manage to get the quality specifications, you can still sell it as feed barley, but you might get 20% less of the price.
Yeah.
There are controls of particularly the partly the pesticides, but also the toxins that might be produced by disease. So there's a test which the farmers can do and the maltsters will do to check that it's pure and not contaminated. And that might mean there are have to be fungicides applied. And because it needs a low level of protein, about 1.7% or less, you've got fertilizer restrictions on the yield. Now that makes it cheaper to grow, but on the other hand, if you're in a soil your farm has a soil with a high level of nitrogen, then you're going to find it hard to get to the required nitrogen percentage. You've got to dry it when it's finished, and if it's a wet harvest period, then that's going to take energy, which is cost. And weeds can get contaminating it, so you've got to not have a great density of weeds there. And the growth will be slow at the beginning, but the grains are produced halfway through and then mature into the harvest. You can clearly see or recognise barley by these awns, sort of feathery ends to the corn. And sowing is, fairly mechanical. Growing, got to manage it, but depends on the climate and the weather conditions. And then maturing, you want the right heat at the right time in the appropriate point in the year. Just before harvesting, checks being made, samples being taken. I mean, nowadays a lot of this, interestingly, is done more remotely as well. There's people looking at drone seeding, so flying a drone over to spray around, drone or remote monitoring of the colour to indicate how the growth is going and the hydration of the plant. Also, when it's harvestable, but it's typically needs a physical presence to take samples to show that they're in the right conditions for harvesting. And the specifications, well, depends on the variety, but there are various approved ones which you would be choosing and are popular and accepted as having the right quality. The viability is very critical. When you harvest the grains, you want them all to be able to germinate. So 98% is very high, only 2% losses. And you can test that in a laboratory condition by putting a petri dish with some water in and seeing how many germinate. The nitrogen or protein content, nitrogen content of 1.7%, protein maybe 12%. That will be testable in a laboratory, and if you go too high, then it would be rejected. So 1.7 will be a good target, maybe 1.8 maximum, down to 1.3. And that's mainly because you don't want too much protein in the wort to precipitate in the beer. The size of grains need to be fitting specification, so it goes through a screening, and any small grains drop through and are used elsewhere, go into food barley. You want the grains to be similar size. You don't want other grain material in. You don't want wheat in. You don't want rye or any other cereals being mixed with them. And it's not impossible that a farmer gets confused or intentionally mixes them to a small degree, but the laboratory will be able to check that quite easily. Don't want molds growing. You don't want the aroma to be contaminated by anything nearby that's been contaminating, and these mycotoxins from mold growth. And also damaged grains as the harvesting goes on. It's a mechanical process. It's not difficult to damage grains in the conveyors and so on. Each year, various varieties are approved, so it takes maybe up to 10 years for a new barley variety to be approved. These are some example ones this last year. I've not tried them myself here, but they will be often more economic for the farmer. Whether they are better for brewing is, a more debatable or negotiable issue. But they might have high levels of enzymes. They might have a coloured spectrum, a flavour spectrum, and so on. And over the years, it's hard to read some of these, but over the years since 2013 to just recently, various varieties decline and various varieties appear. And it's partly fashion, but maybe some varieties are more adapted to climate change conditions. So, you can understand why they are increasing and decreasing. All that said, most malt goes to distillers. Most, a fair amount to brewers, but distillers are the main users. And because the Scotch whisky industry is so big, that's understandable. This is in the UK, though. Internationally, be a different matter. It's worth mentioning heritage barleys. These are ones which have developed over the last century, really, 100 years. Maris Otter and Golden Promise, and they are ones which have particular characters and are traditional. So they would be associated with particular brewing processes, particular brewing styles. And this is some detail about their evolution almost, when they were crossed with this and that and the other. Modern small-scale brewing, more flexible with their sort of malting intentions. But Maris Otter, this one here, it's about 60, 70 years old now. It's increased recently in sales. Worth mentioning heritage malts. Chevalier is an interesting one. Now, this was first grown in over 150 years ago. It was found to be a very big size of grain, when in those days grains were very small. And somebody noticed it and said, oh, I'll grow that again. And it became very prevalent. It was the one that most breweries in the UK and then internationally used because it gave such good quality beer. And it's now been recommercialized. Now, it stopped being produced in around about 1920 because it's a very tall barley. It's about almost a metre tall. And in around 1915, 1920, harvesting became more mechanical. Previously, it would have been scythed and hand processed. But when mechanics came in, it was found to the size of it didn't crop very well. It also blows down easily, more easily if you've got a big field and a large stormy night or something. It'll blow the grain to the ground and therefore reduce the harvest. But we were involved in this where we worked with the John Innes Institute, and we had some research going on to just look at the old grains from the past. And we found in some tests with different fungi that Chevalier was very resistant to head blight. It would not allow Fusarium to grow. It was moderately susceptible to mildew, but once it's very tall, it's away from the ground and the soil. So it's got a very good advantage as being an organic barley, which is therefore good to grow in places which are fairly moist in climate, like the east coast of America. And it's since been redeveloped and is available through CRISP. The big problem is still that it's a tall barley. So in the last few years, we've done some segregants of crossing a short barley with Chevalier to get one which is also resistant and short. And that's, becoming more and more prevalent. So worth looking at. There's other varieties as well, which are in potential with that. So it's, a good thing for the future. But in the long run, what we want is good beer. We want beer which has a high extract from the barley, and the malt doesn't have problems in processing, as you mentioned, lautering and so on. The viscosity is suitable to filter and such. The fermentable wort is to the target specification, but it still has residual dextrins left. The yeast grows well, and it produces the right colour and flavour and everything else we want in a beer, which is the job we have ourselves. Now, brewhouse yield is going to be an issue in that. Fermentation performance, maturation performance, filtration, all of these are processing and product features which our barley and therefore malt provides. And we're going to look at those in other sessions when we talk about the the mashing process, the maturing and conditioning processes and so on to manage that. So really, that's the overview with a bit of a focus on certain areas where we can get our quality beer from quality malt, from quality barley. If all that makes sense.
Yeah.
Good. So let me end show there and return to where we are with all of this. Did it make sense for you, Yvonne?
Yes, because my background is agriculture. So I started off growing barley. I don't do that now. I've progressed into manufacturing, but yeah, so I've got all that background. So that's just revisited a few things for me there. That was good.
Perfect. Excellent. It's one of those industries which, you know, is fairly standardized and hasn't probably changed that much, but the analysis has changed and the varieties will be developed. So, you know, we'll keep an eye on what comes through. But Tassos, if you're taking the exam, then you want to, you know, focus on some of those features which they tend to be more on the knowledge side than the concepts, but, you know, the actual knowing of your processes of the different parts in the malting process and everything, you know, quite important.
Okay.
Good.
That's very good. Thank you.
Thank
So
you.
there will be, well, there is supporting material on the website. Have a go at some of that. There'll be assignments arriving there as well if you want to have a go at some of the material which we give you feedback on, but there are quizzes and tests which will give you automatic feedback, and you can do again so as to perfect your knowledge. Good.
If you do the questions and the quizzes, do you have to do, is it mandatory that you do the assignments as well, Keith?
No, what we say, it yes, to complete the program here on the online, you have to have done the quizzes and the looked at and viewed the appropriate content, and you can see a tick box on your screen which will show when you've done that.
I've seen that. Yeah, I've seen that. So it's not, you don't have to submit the assignments then?
No.
Okay, they're just helpful to do as well if you can do them.
I did the test in about infusion and decoctions, you know, and yeah, I failed a little bit, so I have to read it again. Yeah, because it's the filter press. Yeah, never used that, so it's a little bit.
Yeah, I mean, my experience is we want to give you a range of things that can help you learn and know, but you don't have to be forced down this particular route or that particular route.
Okay.
I haven't looked at the tests yet, Tassos, so it's quite difficult, are they? They're quite difficult.
Yeah.
Yeah, okay.
Yeah.
Thank you.
It is, yeah. Okay.
Okay, well, we'll reserve judgment. Let's see how we go.
Okay. See you.
Okay then, all the best.
All right then, thanks very much. Thank you.
Okay, good to meet you.
Thank
See you
you.
again.
Bye.
Bye.